This study is part of the Tweed project that Tim Smithson spoke about earlier on, which is investigating the evolution of tetrapods after the end Devonian mass extinction. But in this talk, I'm going to talk about the invertebrates and specifically the ostracods and bivalves. And this is because we're interested in actually looking at the whole ecosystem and what repopulated the environment after the extinction, and these are actually the most common faunas that we have present. So just to summarise again about Romer's gap, we've got this gap in the tetrapod fossil record, where at in the late Devonian we've got the occurrence of the more aquatic types of tetrapods, whereas in the later Carboniferous we've got the more terrestrialised forms, and in between a gap in the fossil record. But this project has found numerous new fossil tetrapod material from the Scottish borders and the Midland Valley of Scotland. And as Tim showed, we've also found a lot of rare fish and shark material. And the next slide just is a highlight of some of the fossils, just for you to enjoy. So on the top right is a specimen called Rebo, and a reconstruction of the tetrapod next to it. Unfortunately, we've almost got the entire skeleton, but it doesn't include the head. But it's called Rebo because of the ribs you can see sticking out there. And it's about 1.5 meters long. Many of the other new taxa are much smaller. And another really common vertebrate element are rhizodonts. And here you can see some clethra, which is the bone from the shoulder region. And all of these specimens are from the same bed, showing that there was a really wide diversity of individual sizes within this particular bed. We're also finding a lot of lungfish material and some rare tooth plates, similar to the ones that Tim Smithson spoke about earlier. So just to summarise, I've listed the main fauna there on the side. So we've got the tetrapods, many different types of fishes, a large diversity in arthropods, and the invertebrates and plants as well. What does the microfossil assemblage look like? I've sieved some samples throughout the formation we're studying, and these are the most common elements that you find. We've got a lot of actinopterygian scales, these ones, and next to it is various bone fragments from actinopterygians. The one on the right is part of a lepidotrichia bone, which is part of the ray fin. We've also got rhizodont scales on the bottom left, they're very common. Actinop teeth in the middle, that one, and numerous plant and megaspore fragments, also macroplants, and ostracods as well. I am going to talk a bit about sedimentology in this talk, and I'm going to show you some data from a borehole that we've drilled. But I also wanted to show what the sediments look like in the field, because there you can see some of the more obvious relationships between the different rock types. So this is the field section at Burnmouth, where we've got a 500-metre thick section of rock exposed, which represents approximately 13 million years from the base of the Carboniferous upwards. Here you can see a small channel body which the bag is sitting on, and above that, bearing in mind that all of these rocks are tilted by 90 degrees, above that we've got some very flat-lying units which are resistant to weathering, and these are the cement stones, which are actually dolomitised units. Some of these are diagenetically dolomitised, and some of them we think are original dolomite. But we're looking at a really diverse range of depositional environments in this formation. We've got things like savkas and evaporitic lakes with gypsum and halite forming. We've got more freshwater lakes. We've also got some more saline but not quite evaporitic lakes with the dolomite forming in. We've got some very rare marine incursions that happen. And there's a whole array of different fluvial systems across the area as well. So that's what it looks like in the field. And this is part of the core Just a bit of a comedy slide. We were looking at the Munsell colour chart for paleosols, and another paleosol, because there's 218 paleosols, so yeah, we got slightly sick of them. Don't tell my colleague Tim Kearsey, he's the expert in paleosols. But one of the really fantastic things about drilling this core was the preservation of the sediments. And in the field, we hardly saw anything like this number of paleosols. About 100 were recorded, and desiccation cracks, maybe about 5 from the field section, but from the equivalent thickness of core, 131 desiccation cracks. And that's what I'm really interested in this talk, the relationship between the invertebrates and the drying out of the environment that they were living in, and what that can tell us. So here's the sedimentary log from the core. It's arranged stratigraphically with the oldest sediments at the bottom at 500 metres up to the top. And in yellow are thick sandstone beds. And the rest of these beds are very thin, hence the thin lines. In fact, most of them are too thin to illustrate. The average bed thickness is about 10 to 20 centimetres. And that's because the environment is just changing very rapidly. We're looking at a kind of coastal alluvial interaction here. I've plotted up the occurrence of ostracods, bivalves, fish. I'm looking at the relationships between these three, but also bioturbation. And the reason why I've shown bioturbation is because often the ostracods and bivalves are poorly preserved, so we've got a bit of a taphonomic bias going on. But with the bioturbation, it can tell you that these were sediments where, they were habitable and there might have been fossils preserved, or there could have been. And you can see that where we have got thick sandstones, for example here, there's a gap in the fossil presence-absence because we're not finding these forms living in the sandstones because there were thick fluvial systems coming in that were rapidly deposited, so weren't habitable. Just to convince you that these forms are associated, I've plotted up the percentage of different faunal occurrences with bivalves and with ostracods, and you can see they're commonly associated with fish, and they're associated with each other, so bivalves and ostracods most commonly occur in the same bed. But they also occur with spirorbis, which are microconchid worm tubes that are calcified, and gastropods. Okay, a little bit about the ostracods. I do love ostracods, but I'm only going to show you these few images because I may be the only one here who really likes them. The arrow in green shows the occurrence. So at the top we've got the most common ostracods, and at the bottom the rarer ones. And ostracods are really interesting because they can be used as paleosalinity indicators. And from my previous studies, from my PhD, from the Visayan, I found out that the ostracods that are the most common here have a euryhaline tolerance, so they can be found in brackish and fresh waters. Whereas some of the ones which are rarer are either the very freshwater forms or the more marine forms. But actually, if you look at the whole assemblage, we've got nothing which is shouting stenohaline normal marine conditions. So that's interesting. What are the bivalves? Apologies for this picture. This is your typical bivalve assemblage on a bedding plane. They are usually very poorly preserved, and they're very small. And so I've actually got an image of black and white there of Modiolus from more recent times, just to show you what it looks like. So Modiolus bivalves are the most common, but also Niodytes. This is the first record of it from the Early Carboniferous. And we also have rare occurrences of some more marginal marine forms, which are distinctly different due to their thicker shells and larger size. The really interesting thing about the Modiolus that we're finding is most of the assemblages are only of juveniles, and very rarely do we have adult specimens. We're also getting some in situ preservation. They had a semi-informal mode of life, and we've got some specimens which are preserved in situ as well. So I'm showing the sedimentary log again here, but with some additional information, and we're going to go through parts of this in a bit more detail to look at the fauna. Things that are added on the left is a plot of paleosol thickness, and on the right, next to that, are laminated grey siltstones. And the reason why I'm showing these against each other is they have a fairly nice mirroring effect. So where you have a sequence with thick paleosols, you're not getting thick laminated grey siltstones. And this is because they're formed in sort of opposite depositional environments. Obviously, the paleosols representing a drying out and vegetation of the landscape, the laminated siltstones deposition in water in a whole range of settings, but most commonly in floodplain lakes here. Also, I'm showing roots and desiccation cracks on the right in brown with the dots. And you can see that they're found throughout the formation. We're going to look at the middle part now, which has got a lot of desiccation cracks and roots and paleosols. And what I've done is just expanded that section, 70-metre section, about 100 metres. And I'm showing the bivalve fauna in a bit more detail. The bivalves aren't as common as the ostracods and fish, but they are present. And the most common fish are actinopterygians. Again, throughout, we've got a lot of paleosols and desiccation cracks. But after each of these events, the environment is again recolonised. And now looking at the bottom, where there's less paleosols but greater thickness of siltstone, you can immediately see there's a lot more bivalve fauna, and there's also more ostracods and slightly more fish. We have got less paleosols, but desiccation and rooting is still happening throughout this sequence, despite that. Just to highlight, we've got bivalve other and fish other. These are the more unusual marginal marine bivalves that I mentioned earlier. And in the case of the fish, it's things like rhizodonts, lungfish, and bradyodonts. This is a reconstruction model of the environment at the time of paleosol formation, drawn by my colleague Tim Kearsey. And you can see there's a whole range of different environments, but there's a lot of water bodies that were basically drying out at the time. And how does recolonisation occur? Well, we know from the ostracods that they can have desiccation-resistant eggs, and those eggs can be windblown or they can be transported by water currents. But for the bivalves, it's a bit more difficult because they can't swim. But living Unionidae bivalves actually have a phoretic parasitic larval stage on on the gills of fish. So I'm suggesting, and this is speculation really, just comparing the actinopterygians with some modern fish that actually transport the bivalves, that the same thing might have occurred in the Carboniferous. This is the second to last slide. What about predation and food webs? Actinopterygian jaws and teeth, very delicate and small. They might have been eating the ostracods and bivalves, and there's one record from the Fulden beds by Salon and Coates 2013 of a tooth plate structure in an actinopterygian. But some of the other fish which aren't as common, such as lungfish and bradyodonts, definitely have these shell-crushing tooth plates. And I propose that they were the ones which were eating the more marginal marine bivalves that had much thicker shells. Just to summarise, we are looking at the whole region, and we're looking at the occurrence of the marine and the non-marine faunas, also looking at paleocurrents in terms of the fluvial systems in orange and marine incursions in blue, and this is ongoing future work. So we've got a really interesting assemblage of ostracods, bivalves and fish in these sediments that were later desiccated. I'm proposing a transport mechanism of the bivalves into these environments via the actinopterygian fish. We're investigating what was eating what, and actually we're looking at the whole ecosystem and how it's different in the terrestrial and marine environments that were present. So thank you for listening.